Okay, so this is going to be a quick review of the 3' and 5' ends of DNA structure and how they get numbered that way. And then I'm going to talk about the how to find the leading and the lagging strand and how to do the DNA replication fork practice worksheet. So you guys remember that the sugar phosphate backbone is made of alternating sugars, deoxyribose and phosphate. So it goes phosphate, deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar, phosphate. Lots of students, when asked what makes up the backbone, will say sugar phosphate, but remember, sugar phosphate isn't an actual thing. It's called the sugar phosphate backbone because it's made of both sugar and phosphate. Now, we've talked in class about the 5' and 3' end, and I just wanted to go over how these are numbered. If you remember, deoxyribose is a 5-carbon sugar, and they number them, number the carbons in the ring. Now, these carbons are not being shown in the ring, except for the 5th carbon is being shown, but carbon number 1, the 1' carbon, is the one that's attached to the nitrogenous base. And then it goes 2, 3, 4, and 5. The 5th one is here. The 3 and the 5 carbons are the ones that are attached to the phosphate. So there's a phosphate attached to each 5th carbon and each 3rd carbon. So when we're trying to determine which end of the double helix that we are looking at, if we look at if we're looking at the 5' end, that means that the 5th carbon in the ring, or actually the carbon that's out of the ring, but the 5th carbon number 5 is at that end. And if we look down here, if we count 1, 2, 3, that's the 3rd carbon. And then the 4th carbon would be right here, and then the 5th carbon is here. We care about them because they are anti-parallel, and that has to do with DNA replication. But if we are to, kind of discuss what anti-parallel means, that means that one of them is upside down compared to the other. So if these two pens are like this, they are parallel to each other. If one of them is upside down, they are anti-parallel. Just like if you look at this molecule, on this left side, the oxygens are facing up, and here the oxygens are facing down. So the two strands of DNA are in the opposite directions to each other. So now I'm going to go over how to do the replication fork practice. If this is the 5' end, then this would be the 3' end. And if this is the 3' end, this is the 5' end. Now remember that helicase would be here, and that would be opening up your double helix in that direction. DNA polymerase builds the new strand, and DNA polymerase can only build in the 5' to 3' direction. So this would be our leading strand. So we would label it leading strand. Hopefully you will use better handwriting than I did. And then we would label this the 3' end. Students often forget to label the 3' end of the leading strand in this assignment. And so next, I want to talk about the lagging strand. It's called the lagging strand because it's built slower, because the double helix has to be open for a little bit longer, and then polymerase still works in the 5' to 3' direction, but it has to jump ahead and build backwards. So it builds one here, and this is going to be Okazaki fragment 1, and it builds in a 5' to 3' direction. And then the next Okazaki fragment would be here, and it's built in the 5' to 3' direction, and this is Okazaki fragment 2. And then the next one is built here, 5' to 3', and this is Okazaki fragment 3. A couple things to keep in mind. Students often misnumber these and think this is the first Okazaki fragment, but if the strand is opening this way, this end has been open the longest. So it opens a little bit, builds a fragment, opens a little bit more, jumps ahead and builds a fragment, opens a little bit more, jumps ahead and builds a fragment backwards. These are called Okazaki fragments because of the scientist who figured out what was going on this strand, and this strand is referred to as the lagging strand because it lags behind. It is built more slowly. When you do this, you do not need to use different colors. You can use a regular pencil to do this. Keeping things in mind, label your leading and the lagging strand on the inside because these are the new strands being built, and this one is being built faster, so this one's in the lead. This strand is being built slower, so this one's in the lead. If you label it out here, that doesn't make sense because those strands are already built, so they can't be in the lead or lagging behind. Students also, in addition to forgetting to, or getting the Okazaki fragments labeled in the reverse, not knowing it goes 1, 2, and 3, they sometimes forget to label the 5' and the 3' end of every fragment. And so the reason I did these in different colors is so you'd be able to see the red 3 here and the blue 5 there to remember, and like the purple 3 here and the red 5 here. Most people remember this label and this label, but do not get the labels in the middle here correctly. So then your job is to do the other 3 forks on your worksheet. This one is the sample. And then you do these 3, and you can either turn it in class, or if you're doing this from home because you were absent, you can take a picture of this and upload it with the DNA replication review.